at high resolution in adapting bacterial populations. So before Michael can start, maybe I can remind you that you are most welcome to ask your questions in the Zoom chat during the talks. And at the end of the talk, we are going to read the questions, or you can even intervene by unmuting to ask your own question. Okay, what else? I mean, yeah. Whether you want to leave your camera on and off is up to you. Leaving it on is good for the speaker to see some faces. Putting it off is better for the bandwidth, so up to you. And so now I think we can start quite soon. It's a bit early, so I mean, I don't want people to, yeah, those who wanted to just join for the talk maybe still coming. 2 minutes early. So Michael, are you able to share your screen?
Yes, I think so.
Okay, that's great. Hi, welcome.
Hello, thanks to everyone, for the organizers and everyone for attending virtually.
And thank you very much for being our first speaker. That's, quite an important task, right? Especially if there are any technical issues, you will be, first time, but I hope not. Okay, we can see your screen now. It's not in presentation mode yet, but it's visible. Okay, I think it's perfect this way.
Now you see the full slide.
Yes, I see the full slide. Yeah, and it's progressing well. So I think all is technically working, which is great.
Should I wait another minute or should I?
I mean, it's 29, so I think, yeah, we can wait a few seconds and then you can start. Okay.
I think we can start.
Okay, yeah, I mean, that's fine. I was just worried about, some late minute people, but I think you're right, it's not increasing that much. So Michael, we are very happy to hear your talk. Yeah, feel free to start.
Well, thanks again to all the many organizers who put this together and to everyone for attending. It's really nice to be here, albeit virtually. As Anne-Flo said in her introduction, my name is Michael Manhart. I'm at the Institute for Integrative Biology at ETH Zurich, and I want to tell you about work we've done over the last few years on tracking lineage diversity at high resolution and adapting bacterial populations. So this project has been a collaboration with the groups of Shimon Burstein and two of his students, Veronica Jasinska and Jesse Lerner at Ben Gurion University in Israel, as well as the group of Adrian Sarijos and his student Louis Gauthier from the University of Montreal. So as I think probably everyone here appreciates, whole genome sequencing has become an extremely powerful tool for tracking very quantitatively the population and evolutionary dynamics of populations, and especially microbial populations. But I think it's important for us to also appreciate what kinds of things whole genome sequencing can miss when we try to understand these populations. And so what I mean by that, I think can be best understood by looking at this spectrum of population sizes measured as number of cells on a logarithmic scale, as I've shown here. And so, for instance, a typical laboratory population will be somewhere between, say, 1 million to 1 billion cells. But when we perform whole genome sequencing on these populations, we're generally only able to detect variants present in these populations at about, say, 1%, maybe 0.1% or so. And if we translate that to actual numbers of cells, that would correspond to something about 10,000 cells. And this is important because we have to always remember that when new mutations arise in these populations, they always arise down here on a single cell. And what this means now is there's this gap in the space of frequencies that we're not able to detect with whole genome sequencing, in this case, 4 orders of magnitude. So therefore, there's a lot of possible dynamics that are happening in populations that we're completely blind to with whole genome sequencing. And then if we extend this to thinking about natural microbial populations, so for instance, the human gut microbiome, those tend to be much larger actually, say 10 to the 13, 10 to the 14 cells. And this limit is even more problematic because it's still a relative frequency that whole genome sequencing can detect, 1%, and that leads to an even bigger gap in frequencies that we're therefore blind to with this approach. So an alternative that addresses this limitation of whole genome sequencing for understanding population dynamics is using something called DNA barcodes. And DNA barcodes are just short sequence labels that we can use to track lineages in a population. And so the way this works is that we start with some library of barcode sequences, schematically shown here, and these could either be design sequences or in our case, they're just random sequences. And we want to insert each one of these sequences into the chromosome of a microbial cell in our population. And then as these cells grow and divide, because the labels are on the chromosomes, they're passed down to the descendants. And therefore, by sequencing these labels at different time points, we can track how these initially labeled lineages increase or decrease in frequency over time. And because we're using sequences here, we can generate huge amounts of diversity in these libraries and track extremely large numbers of lineages at a time. And because we can concentrate all of our sequencing depth on these single labels, we're able to detect very low frequencies in populations down to say 10 to the -6 or so. And that's really where we gain this advantage relative to whole genome sequencing. So this is an approach that's gained a lot of different applications over the last several years in many different organisms. But up until recently, approaches for doing this in bacteria were relatively limited in terms of the number of unique barcodes that were possible. Generally, these approaches were able to achieve maybe a few hundred to maybe a few thousand unique barcodes. And so a few years ago, Shimon and Adrian and I teamed up to try to address this problem. And we developed this approach that's able to generate very high diversity DNA barcode libraries in bacteria that are orders of magnitude higher diversity than what was previously available. So I'm happy to discuss technical details of the method later with anyone who's interested, but suffice to say it relies on a transposon called TN7, and we're able to generate about 500,000 unique barcodes in E. coli. But what's more, this method, because of the TN7 machinery, is very generalizable to many other bacterial species as well. And that's something we're actively working on now. So we really see this as a very generic tool for generating these kinds of libraries in bacterial systems. But with this library in E. coli we wanted to see what was something really powerful we could do with this that we couldn't do in any other way. And so, like I said, you're able to generate, you have a very large number of these barcode labels you can track simultaneously. And because of sequencing, we're able to detect very low frequency lineages in the population. And so one of the places where that kind of approach is most valuable is when you have a situation of very weak selection and very short time scales. And a scenario where we thought this would be most important would be evolution under very low concentrations of antibiotics. So obviously, evolution of resistance to antibiotics at high concentrations has been a major topic of research for many years now, and we know a lot about how populations evolve resistance and evolve more generally under those kind of conditions. But very low concentrations of antibiotics are believed to be present in many natural and clinical environments, inadvertently, of course. But we know a lot less about how those kinds of environments with these very low concentrations of antibiotics can affect evolution of bacterial populations. In particular, how, what are sort of the lowest concentrations where there still is an effect on evolution. And so this is, again, a situation where we have very weak selection. We expect a very large number of simultaneously segregating beneficial mutations. And so this is where having a barcoded library can be really valuable for for understanding more of the details about what's actually happening in this scenario. So we focused on two common antibiotics in this case, chloramphenicol, which I'll abbreviate as CMP and denote by this blue color here, and trimethoprim, which I'll abbreviate as TMP and denote with this red color here. And we considered two different concentrations of each of these antibiotics. One that I'll call low, and that's the darker version of the color, and then another that I'll call ultra low, and that's the lighter of the two colors. And as you can see, both of these concentrations are just small fractions of the minimal inhibitory concentration, or MIC. So we expect both of these to be quite weak. And finally, of course, we have a fifth control environment where there's no antibiotic at all. And the experiment we did was to take this bar-coded E. coli population and evolve it under serial passaging for 70 times, which corresponds to about 420 generations of growth. So the first thing we wanted to look at here was what kind of resistance in the population evolves under these different antibiotic conditions. So first we can look at resistance to chloramphenicol, which I'm measuring as the IC50 on the vertical axis here. And we can look at how this changes over time of the evolution experiment. And we see that the populations in low chloramphenicol in dark blue here, those evolve significantly greater resistance to chloramphenicol over time of the experiment. In contrast, the populations in ultra low chloramphenicol don't seem to change in any significant way. And in fact, you can't even see the lines here because they're just overlapping with the control populations and no antibiotic. And we see something qualitatively very similar in trimethoprim, significant adaptation in the low condition and negligible adaptation in ultra low condition. And so what this suggests is that maybe these ultra low concentrations of the two antibiotics are so low that maybe they just have no effect if we don't even see resistance evolving at the population level. But now, since we have this population bar-coded, we can ask whether that's really true if we now sort of look under the hood and look at these really low frequency lineages in the population. And so we can sequence the barcodes in each of these populations at several different time points and put together all of these lineage trajectories that way. So what I'm showing here is basically the sum of all this data that we get from this sequencing. So each column here represents one of the 5 environments, and each row is a different replicate population in that environment. So we have 3 replicates in each environment. And each panel shows the frequency dynamics of all 500,000 bar-coded lineages in that population. So there's of course a lot of information here, but I think you can glean some of the most salient features of this experiment just by looking at these kinds of plots. And so, for instance, one of the most important things that we can observe is that in just about all of these populations, one or a handful of lineages dominate the population by the end of the experiment. And a simple back of the envelope calculation would indicate that that would not be possible purely from neutral processes. And so that means that selection is really the driving force here for the changes in lineage frequencies that we see. And in particular, selection for the particular antibiotic condition that is used in that environment. And the other thing we see is that if we look at replicate populations within an environment, say the 3 replicates in low chloramphenicol in this first column, we see that they look relatively similar in terms of their overall dynamics. Whereas if we then look at populations in different environments, say the low chloramphenicol replicates to these ultra low trimethoprim ones, we see systematic differences. So we'd like to take a more quantitative approach to understanding these observations. And so for this, we can take a page from ecologists and think about how the diversity of lineages in these populations not only changes over time within a population, but also compares between populations. And this approach, I think, is really powerful in this regard because, for the same reasons it's so useful in ecology, it's really very generic and generalizable to a wide variety of different environments and sort of evolutionary situations where you might have changing environments or migration. And so I think this is a really flexible approach for understanding this kind of data. And I would like to highlight a few of the insights we can gain from this. So if we want to quantify lineage diversity, how can we do that? Well, imagine our population is this little cartoon box here, and we want to quantify the diversity in this population. And we see there are red cells, there are blue cells, there are green cells, there are purple cells, and orange cells. So we might say, well, there's 5 types there, so the diversity of this population is 5. But you might also wonder, well, this orange type is only present in one cell, whereas red is present in half the population approximately. And so maybe it seems a little misleading to count those both equally towards diversity. And this is in fact something that's captured by this concept from ecology called the diversity index, which for the physicists here is equivalent to the exponential of the reigning entropy, which may not be surprising. And this diversity index is defined as shown here, and it's basically an average over all the lineages K in the population weighed by their frequencies xk, with this additional tuning parameter called Q. And this Q represents the relative weight on these low frequency versus high frequency lineages in the population along the lines of what I just sort of illustrated at the beginning of this example. And so when Q, for instance, is low, so in particular when Q equals 0, this puts more weight on low frequencies. So when Q equals 0, this just counts the number of lineages in the population, like I initially said, irrespective of their frequencies. And so in ecology, this was the same as what would be called species richness. And then as we increase Q, that gradually puts more weight on higher and higher frequencies in the population. So when Q equals 1, this would be called Shannon diversity, which is the exponential Shannon entropy. And then as Q goes all the way to infinity, then this diversity will depend only on the lineage with highest frequency in the population. So now we can use this to really capture not only how the diversity of the population sort of compares, not at different sort of scales of frequency. So we can calculate this for all of the populations in our experiment. And so what I'm showing here is the diversity index for each of these 15 populations as a function of time in the evolution experiment. And first I'm showing it to you where I've set Q equal to 0. So there's more weight on low frequency lineages. We're just counting unique barcodes in this case. And as you can see, this really leads to a very sort of clean and reproducible set of dynamics for this data. And that actually kind of recapitulates these observations I made a few slides ago. So we see that the different replicate populations for each of these environments, say the low chloramphenicol environments, they all follow a very similar pattern of losing diversity during time of the experiment. And then there are systematic differences between those dynamics between each of these environments. So low chloramphenicol loses diversity most quickly, followed by low trimethoprim, and so on. And one of the most other striking things that we see when we look at the data in this way now is that the environment of ultra-low trimethoprim actually loses diversity more slowly than even the environment with no antibiotic at all. This is kind of a tangential story to what I want to talk about today, but I'm happy to discuss that further with anyone who's interested. And I think one of the important things to also point out here is that using simulations of models, we can show how this rate of diversity loss, as shown here, is related to the speed of adaptation in these populations. And so this is really kind of a proxy for how adaptation is proceeding at these different speeds in these different antibiotic conditions. So like I said, we can also look at this at sort of different bands of frequencies by varying this Q value. And for instance, if we set Q equals 1, we put more weight on high frequencies, and we can see how the dynamics would change. And in this case, they're qualitatively quite similar. So that's diversity within populations over time, but we'd also like to understand how diversity compares between populations. And in particular, we'd like to understand diversity between replicate populations in the same environment to understand how predictable evolution is at the scale of these individual lineages. And so to do this, I need to introduce one other quantity that I'll call dissimilarity. And this is defined such that if we have two populations that have identical diversity, so the same lineages at the same frequencies, they would have dissimilarity of 0. And if we have two populations with completely non-overlapping diversity, we would have dissimilarity of 1. And this dissimilarity is basically calculated as the ratio of the diversity of the populations to compare pooled together divided by the mean population diversity with some shifting and rescaling. And this is what ecologists would call beta diversity. So we can now calculate this for all the replicate populations within each of these environments. So here on the left, I'm showing that dissimilarity between those replicates for each of the 5 environments. And so at time 0, the dissimilarity starts at 0 because the populations are identical. And then you see that quickly the populations in each environment start diverging from each other and they become more different. And they do so at these sort of time scales that are characteristic of adaptation in each of the environments, just as we saw when looking at the diversities of the individual populations. So this is, if we look at that dissimilarity at sort of the low frequency lineages. But what happens if we look at it for the high frequency lineages? Well, for 4 of the environments, we see something that's qualitatively similar, this sort of pattern of divergence. But if we look at the replicate populations in low chloramphenicol in dark blue here, we see that they do diverge at the beginning at high frequencies, but then around generation 150, they turn around and start converging. And the high frequency lineages in these populations actually start to become more similar to each other, until by the end of the experiment, they're actually about 80% similar. So why is that? What's going on with that condition compared to these other ones? And so again, we can take advantage of some unique insight afforded to us by this barcoding data, and we can look at the individual frequency trajectories of these lineages. And if we look at the adaptive lineages in these populations, we see that they generally fall into one of 2 qualitatively distinct classes. So one class looks like the green ones here. So these are lineages that increase in frequency right away at the beginning of the experiment, indicating that they contain some pre-existing beneficial variation on them. Whereas lineages that follow the orange trajectory pattern on the right, these are trajectories that don't increase right away, but then increase later on in the experiment. And that suggests that these are lineages that acquire mutations, beneficial mutations de novo during the experiment. And we can systematically identify lineages of both types in each population by performing hierarchical clustering on these different trajectories. So just to convince you that this works very well, this is an example of the correlation matrix of these adaptive trajectories from one population, and you can just see that the clustering structure is sort of very robust. And so we can use this to then really quantify the relative contributions of these 2 sources of variation, pre-existing and de novo mutations in each population. So what I'm showing here now is the total of all the lineage trajectories in each population, separated according to which ones seem to contain pre-existing mutations versus which ones seem to contain de novo mutations. And this therefore really directly explains this property of the low chloramphenicol condition, because we see that low chloramphenicol here is completely dominated by these lineages with pre-existing mutations. That set of lineages basically fixes within the first 100 generations. And as a result, therefore, it's going to be the same lineages rising to high frequency over and over again because they had some beneficial mutation that was pre-existing that drove their initial adaptation. And so that's why we see this convergence of those high frequency lineages in those environments. Whereas in basically all the other environments, this pre-existing variation is certainly important at the beginning in driving adaptation, but then later on it gets sort of outcompeted by these de novo mutations. And therefore we see this divergence of lineages in all the other environments. So with that, I'd like to just briefly summarize the main messages that I hope everyone can take away from this talk. So first, I want to emphasize how these different antibiotic conditions lead to distinct and reproducible patterns of lineage diversity loss. And in particular, these ultra low concentrations of antibiotics can produce these distinct signatures in lineage dynamics compared to no antibiotics, even when there's no noticeable resistance at the population level. And this is something we can really only detect with this sort of high resolution barcoding approach. And one aspect of that that I think is most interesting is the fact that this ultra-low trimethoprim condition actually has the unexpected effect of slowing adaptation compared to no antibiotic at all. And finally, we were also able to use this data to quantify the relative contributions of pre-existing and de novo beneficial mutations. And we find that both sources of variation are certainly important, but there are differences between the two antibiotic conditions in terms of the relative contributions. In particular, we see that chloramphenicol depends more strongly on these pre-existing mutations, which is consistent with what we know about from previous studies of adaptation in chloramphenicol, where there tends to be a much broader spectrum of adaptive mutations compared to trimethoprim. So with that, thanks everyone for your attention, and I'm happy to discuss anything further.
So thank you very much, Michael, for the great talk. So again, we welcome questions